Hello, everyone. How are you doing? All right. So my name is Kimberly Ong, and I'm with Virio Advisors, and I am so pleased to be part of this amazing lineup of speakers, and it's been so great to be here and get to share ideas. I've had so many good conversations, and I feel super inspired. Yeah, I am a toxicologist, I'm a biologist, and today I'm going to talk about safety. Yay! Yeah, okay. All right. So, you may be wondering, what on earth is a safety person doing here up on stage? This isn't a regulatory session. Well, I'm here to tell you that safety is beyond regulatory considerations. And in fact, it actually is a lot about sustainability as well. So we think about the three kind of like old school pillars of sustainability. Of course, we want to think about the safety of our products, make sure that the inputs, make sure that the products themselves are safe when they get disposed of, if there's any waste coming from your system, making sure they're not affecting anything in the environment. From a social perspective, of course, you want to make sure that your employees are safe when they're actually manufacturing your products. And you want to make sure that the groups in which you're providing your products to are benefiting from it, that they're safe, that they're healthy, and maybe they're actually advantageous to them. From an economic standpoint, there is, of course, a lot of risk to not being safe. So if your product is not safe, you won't get through regulatory review, which means that you can't put a product onto the market. That's going to be an issue. If your product gets onto the market and someone gets sick from it, for example, that, of course, is going to affect your brand. So, yeah, hopefully I've convinced you that safety is important and part of this session. All right, so why do you want to assess safety? Okay. Uncertainty creates the risk of negative perception for new products. And this uncertainty can also lead to bigger issues. So for example, the negative perception of the safety of GMOs is often conflated with things like unethical business practices and monopolies. And so what you want to do is you want to build confidence among your stakeholders. And one of those solutions is to proactively demonstrate safety. So who are these stakeholders that could affect your safety story? Of course, you have the regulatory agencies. They're the ones who approve or deny your products based on the basis of safety. And they also release guidance and opinions, recommendations that can also help you standardize the way you approach your safety testing and the types of data that you're supposed to submit. So they can really speed up the process when that kind of stuff comes out. And of course, they provide their own independent research, which ends up in the public domain, which can be helpful for you. And then we have NGO, nonprofits, and advocacy groups. We have a couple wonderful ones in this space. We have New Harvest, we have GFI. They're providing a lot of great safety information, good data, things that you can work off of. However, if there's any sort of doubt about the credibility of your safety or sustainability claims, you can also have groups that work against you. You can have groups that, for example, do their own independent studies, create their own reports and opinions with negative aspects of data. They can even file lawsuits against you or the regulatory agencies who have approved your products. They can start petitions. And so basically, they can try to influence the public and policymakers to reject your products. Then, of course, we have the public. They have enormous power to share opinions over social media. So if there's any sort of doubts about your product, you may see them there. And of course, they have purchasing power. The media, of course, provides information to a wide audience. And so even kind of the headlines that get put out there can really affect how the public perceives your product. And then we have academia. So academia is amazing to work with in a safety context. They can be really helpful early on to develop methods and ways to kind of provide some data for you. But I do want to highlight that in academia, there's also a real incentive to publish interesting results. And unfortunately, in the toxicology world that I'm from, the incentive is to publish results that show adverse effects, so things that are actually harmful about your product. So you really want to work together with academia in order to provide product, let them understand the conditions of use, and kind of work together with them. And then finally, of course, there's industry. So it's in your power to be transparent, decide how much data you want out there, be proactive in your testing, and gain that consumer confidence. And I also want to mention that as an industry, If there's something that gets put out there that ruins the trust in your company or in your product, it could also affect the rest of the industry and your peers as well. So I want to tell just a little story about titanium dioxide, just to kind of highlight the importance of really understanding your product and being proactive about safety before it goes to market. So titanium dioxide is a whitening agent that's been used for over 40 years. It goes into cakes and gum. It goes into like Twinkies and mayonnaise and all kinds of different products just to make it really nice white that consumers like. So it's been approved since 1966 in the US, and it's been approved since 2008 in EU. And around 2011 or 2012, there started being some safety concerns that came up because groups were starting to really look at these particles. And what they found that there were like little bits of nanomaterials in there. And these nanomaterials caused some concern because there wasn't enough data on these particles. So advocacy groups picked this up. They started testing a number of different products, taking candies and confectionaries. And then in the case of Dunkin' Donuts, they looked at the powdered sugar on these donuts. And they did actually find some fractions of these nanoscale titanium dioxide particles in the powdered sugar. So they put together reports and delivered them directly to the shareholders of Dunkin' Donuts. These shareholders then decided to push back, and Dunkin' Donuts eventually ended up having to take the titanium dioxide out of their products, even though it was very cost-effective and very efficient product. And technically, at that point, it was still legal. In the EU, a number of other companies followed suit because of public pressure and have banned TiO2 in a number of their products. And then recently, titanium dioxide in the EU is no longer considered safe when used as a food additive. So after 40 years of being on the market, it is no longer allowed to be used. Now, I'm not saying this to scare you or be hesitant about going to the market, but it's basically just a story just to say, make sure you really understand your product, make sure you're proactive about safety, and have that data available. All right. So, demonstrating safety can be challenging. There are a couple challenges here. So for all novel products, currently they're being reviewed on a case-by-case approach. So you aren't necessarily sure what kind of data you need to be submitting. And the onus is on you as a manufacturer to determine what the novel risks are. So taking a look at what kind of inputs are going to your process, maybe how those are being metabolized or new things happening in your product, and determining if there's anything in the final product that could be of concern to the environment or to consumers. Furthermore, because these are whole foods, as Larissa mentioned earlier, new or adapted testing methods may be required. In our experience, we can go to commercial labs and get some great data, but when you need kind of these new or modified testing, it can be a real challenge to be able to work with that in a commercial lab that's used to following really typical standards. This is where academic labs come in. They can be super helpful to develop these methods. However, in a regulatory context, sometimes these labs, the data that comes from these labs can't be used because they don't meet certain accreditation, for example, good laboratory practice. So this is where I just want to do a little plug for Virio Advisors because these are the kind of challenges that we meet. Vireo was established in 2013, and we're a small group of safety advisors. And we work with partners to bring better and safer technologies to market. So we're a small, mission-driven firm, and we work in emerging markets because we truly believe that the products that you all are making are going to change the world, and we want to help the safe innovation and commercialization of these products. So we help organizations navigate the environmental health and safety landscape, working with small groups who are maybe early in the research and development stage and want to think about all the potential challenges that be coming up ahead and perhaps producing their products in a safe way early on, right up to groups who are ready to commercialize and go through regulatory review and need to put together some safety data. Now, the best thing I like about my job is that we get to build and work with consortia. So we try to work with academia, industry groups, competitors, bring them together in order to develop safety data methods pre-commercially, so that when you're ready to go to commercial stages and regulatory stages, all that data is there and available for you. So this brings me to the safety initiative. So this cell-cultured meat and seafood safety initiative that you've heard of quite a few times was done in collaboration with New Harvest in 2020. And it was an industry-wide initiative designed to address a major gap. We realized there wasn't enough publicly available data out there, and we wanted to help guide what kind of research priorities there would be. We were hoping that some groups would take on some of these research priorities so that they would generate some data for the industry to use. So for the initiative, there were over 87 individuals from 50 companies, which at the time was estimated to be about 61% of the industry. And we also gathered input from researchers and various safety experts and food experts in the field. We brought together these different groups in 3 virtual workshops, and also Isha Datar of New Harvest did a lot of one-on-one engagement to talk to different companies. One of the unique things about this initiative is that we did not sign any NDAs, but we still wanted to protect the intellectual property of all the different companies. So we used these different approaches, plus we sent out a number of white papers that we asked for comments and review from all these different experts and participants that they could comment on, and they could do that anonymously if they wanted. And as part of this safety initiative, we have published this paper. It is available in comprehensive reviews in food science and food safety. This came out a few weeks ago. And I just want to quickly acknowledge my co-authors, Jeremiah Johnson and Isha from New Harvest, Vince from IFF, Dwayne, who at the time was working with Mosa Meats, and Joanne Shacken, the president of Vireo. Because, yeah, this was an enormous effort. There was a lot of data to work from, and it was pretty fun to write. But I do want to send everyone here because there's a lot of information here, and I'm only going to just really give a general overview of everything today. So the first thing we wanted to do is think about what we could learn from other fields. So of course, we don't want to reinvent the wheel every time we think about the safety methods that we're going to use. And so we took a lot of approaches from conventional food and feed areas. We looked at also biotech and pharma and medical areas as well. And then we thought about how we could use our practices for safe manufacture, how we could use methods that are used for safety assessment of the different inputs, and also methods to evaluate the final product. So starting with practices for safe manufacture, there's a number of hazard management systems that are already out there. So these are ways to make sure that you're producing things safely. And just starting with good manufacturing practices, GMPs. These are systems that apply to all foods already. They're the general good housekeeping practices that you use while you're manufacturing. So things like making sure you're working in sterile conditions, making sure that you're documenting everything and having appropriate training amongst the people working with your foods. So yeah, these are already applicable, and they can definitely be applied to this field and are often a regulatory requirement in most jurisdictions. Of course, this field's a little bit unique in that you work with cells and have some in vitro things to think about. So we look to good cell culture practice, and indeed you can pull some of those aspects related to in vitro work and sterility and cell media and so forth. On the other end, you're also taking biopsies in some cases from livestock or from animals. And so things like good agricultural practices can give some guidance on how to approach the biopsies and animal welfare. And then good hygiene practices are about handling food, and those are also applicable. One of the things that we really took a look at were the hazard analysis and critical control points. HACCPs. This is also a requirement for a lot of regulatory jurisdictions. And basically, it's a systematic review of the way you manufacture things and trying to figure out at each stage where there could be hazards introduced. So biological, chemical, or physical hazards, and putting into place a control to make sure that that hazard is controlled, or you would be able to catch that hazard if indeed it happened, like microbiological contamination. So the paper that we wrote was intended to help companies start off doing their own tailored HACCP plans. And in fact, New Harvest is working on an online tool based on this paper to help with this development. Okay, so we're looking at the assessments of inputs next. We had to start from a baseline as part of the safety initiative. And what we tried to do first was to develop a generalized understanding of what the manufacturing process was. And so this is what we came up with at the end of the safety initiative. The colors are not coming up properly here, but that's okay. You can take a look at the review paper. It's not important right now. I'm not going to go through this in depth because I know that everyone here is already very familiar with the process, but I just wanted to introduce this because this is, again, a tool that we're developing online for companies to use. So this was a generalized manufacturing diagram using a descriptive, not prescriptive approach. So basically not telling everyone what kind of terminology to use, but just using very descriptive terminology. So we had the core activities. So these are activities that a lot of companies would use. Sometimes they use some of them, not all of them. They can go in different orders. You can see they're like puzzle pieces. You can move them around. And it goes basically from procurement all the way down to harvesting and food processing. Then we have our modifier steps. These are steps that give you a little more information about the different core activities, and they may or may not be used in different processes. So we have things like genetic modification, use of inherent surface like scaffold or microcarriers, and we may have like options like you may take a sample from a slaughtered animal or a biopsy from a live animal. And then we had the targets, which are basically the inputs and outputs of all the various steps. So as I mentioned before, this is kind of how it's envisioned to be used. This is just a fictional example. So companies can kind of take these pieces and move them around to develop their own diagram. And basically what we did with this was we took all these different steps and we tried to identify all the different hazards that could happen in each of these steps. So these were kind of the key hazard considerations that we came up with each of the steps. And I'm not going to go over every single one today, but I will talk about some of the key ones. I just wanted to highlight that there are some things that come up in every single step, like microbiological contamination. And there are some things that are specific just like one stage. So for example, cryoprotectants, you need to consider how hazardous that is during cell storage or cell banking. Okay, so quickly to go over some of the hazards. You of course may have some hazards that arise from the source animal. We know that fecal contamination is probably not going to be a major issue in cell cultured meat and seafoods. However, there is a potential to take biopsies from animals that could transmit infectious disease agents, so things like viruses, parasites, and prions. There are some ways to prevent these hazards. So for example, sourcing animals from pathogen-free closed herds or flocks, so ones that are already used for conventional livestock and that already have really well-defined health screening programs. However, we do recognize that there will be samples taken from some animals that may not be under closed conditions, so things like marine life or exotic or wild animals, and so maybe some additional screening or testing will need to be done on those cells. And there are some tissues that do tend to harbor certain kinds of infectious disease. So for example, if you think about prions, they can be in brain tissue or spinal cord tissue, so you may want to avoid using those. And from what I understand, that's not commonly used in this field. All right, I'm not going to talk too much about microbiological contamination, because if you're in there working with these cells, you know a lot about it. Just going to say that mycoplasma are one of the main concerns, just because these are trickier to detect. And they can affect a pretty high percent of cell lines. But there are ways to prevent these hazards. So when the cells come in from outside the lab, you may want to quarantine any new cells. You can use inactivation methods in order to destroy some of these microbes. And also through your regular process monitoring, you're probably going to be able to monitor for any sort of changes in your cells and be able to find some of these contaminants. So the cell culture reagents themselves kind of course, introduce some issues. Some of the main ones are the animal source components, so things like trypsin and amino acid, and particularly fetal bovine serum. There are some reports saying that up to 50% of commercially available FBS actually contains viruses. We know that people are moving towards synthetic, kind of more tailored cell media. So some of the concerns there could be any sort of novel substances or allergens, things that have never been used previously in foods. And there may be some increased concentration of certain components, things that are already present in our foods, but maybe you will see higher concentrations of them. So maybe we'll see some growth factors, organoleptic additives that are added to food to I don't know, change the pigment or something like that. And we know that some groups are looking at recycling of this media. So we know as the media is recycled, you may actually have an increased concentration of certain components as well. And then something I get asked about often are antibiotics. We obviously don't want to overuse antibiotics. They do, we don't want to contribute to resistance. We don't want antibiotics to be in our final product as well. But again, it seems like the field has generally agreed to decrease or limit the use of antibiotics, and if they are used, they're generally used near the beginning stages of the process, so they wouldn't end up in the final product. So in order to kind of prevent some of these hazards, it would be recommended to isolate animal-derived components from pathogen-free animals, or even better, don't use animal-sourced components. Again, you can inactivate the media, and in your cell culture media, you can consider using only food-grade substances, so substances that are already authorized for FDA use, and also meet certain specifications to make sure they're pure enough for food. All right, I'm just going to briefly touch on genetic modification. We know there are, of course, some intentional approaches to genetic modification, but then there's also things like genetic drift to be concerned about. So with subculturing with the cells over time, you may get some changes. And some of the potential hazards that we would be concerned about would be the alteration of protein expression levels, any expression of novel products. So those could be things that aren't normally present in food, removal of nutrients, production of anti-nutrients, and of course, the production of any allergens could be an issue. Also, if you have any off-target mutations, you may get some genomic instability, and again, some different expression levels as well. And of course, there's a number of different inputs that we're aware of that go into this process, cryoprotectants, scaffolds, some of the things related to scaffolds, like the cross-linking agents, dissociation agents to remove them from the scaffold, and then other chemical substances that may be unintended. So things like leachates from packaging, cleaning products that are used in the lab or in the manufacturing process, and other things that we may need to look for. And of course, it's conceivable that novel substances may be used. So how do we demonstrate safety? There's a few things we need to think about. We need to think about the inputs that go into the system. We need to think about the residues that could be on that final product or in the final product. So they're not intended to be in there. And then we need to think about how we can demonstrate the safety of the full product itself. So in terms of testing inputs, these are typically pretty well-defined substances, and we already have a system for doing that. That's how most regulatory systems are set up. So yeah, we have lots of standard test methods already to do this kind of testing. The one thing I do want to highlight is that typically for novel inputs, one of the approaches to safety testing is an animal test. There is a 90-day subchronic rat study that is sometimes a requirement in order to get authorization. And of course, in a field where we're trying to avoid animal use, we may need to think about some alternative approaches to this. There are no validated approaches to this yet. In terms of the residues and the other things that could be on the product, there already are lists of impurity or residue levels for conventional products. There are lists of authorized antibiotics used in meats, for example. Of course, for cell-cultured meats, we may need to think about standardizing these lists or adding some new chemicals and substances for this field. Yeah, we already have a baseline to start from. And also, there already are pretty standardized methods to measure the residues, so don't need to reinvent the wheel here. Finally, we're thinking about the final product. So the first generation of products are likely going to be designed to mimic or be quite similar to existing products. So we might be able to look at the composition, nutrition, and functional equivalency to conventional foods. And if they're equivalent, then we might be able to rely on the existing safety data for that food. However, if there are any differences, like if there's any novel proteins or allergens expressed, that might direct further safety testing. So some of the challenges we foresee in this field is determining what exactly does equivalent mean, so when can we rely on that data from the conventional food? And also, Larissa mentioned this yesterday, and it's something that's been on our mind for quite a while, is how on earth are we going to do whole food testing if that's necessary? If we want to do in vitro tests, that means we need to like solubilize the product in order to do in vitro testing. And again, if we're talking about in vivo testing, which we really want to avoid, I'm not really sure how we're going to feed meat or seafood to rats. So I'm not going to go into depth about all the research priorities that came out of the paper. There were 19. And so this is just kind of an overview of the different areas. Basically, we established that, yeah, we have a lot of baseline to work from already. We don't need to reinvent this whole field of safety testing for cell cultured meats. However, we do need to validate some of our assumptions. For example, are these products in fact going to have less contamination than conventional products? That's something I hear get thrown around quite a bit. We may want to develop some industry-wide standards for safe residue levels. And in terms of testing, we need to think about the questions about equivalency and also developing some animal-free safety testing methods. So finally, what can you do? Well, you can work with academic partners and safety experts in order to proactively demonstrate safety. One thing I really want to kind of harp on here is some collaboration. So we talk about collaboration a lot, and safety is actually a really good area where companies can work together. In our experience, this is really good in a pre-commercial way, so we can bring companies together, find out what their needs are, and develop these methods and safety tests so that when you get to that stage where you need to provide data for your product, those are already in place. And so this can really be more efficient, it's more cost efficient, more time efficient, and is a way for all of us to work together. So this is something that I've been working on in us at Vireo. We would love to bring together a group of industry, of researchers, of academics, and people, and different stakeholders to really talk about how we can do this together in a collaborative way. So if you're interested in this, please come find me afterwards or email me, because I think that'd be a really exciting project to do together. Documenting everything that you do is super important. I say this because, of course, traceability purposes, but also perhaps when you go to regulatory agencies in a couple of years, they may ask for, let's say, data on your source animal. And you may have collected those cells like, I don't know, 2 years ago, 1 year ago. And so you need to have everything in place so that you can reach back and understand where all your processes came from. Stay up to date with regulation and emerging regulatory topics. Be active in commenting and policy, regulation development, standards development. And, of course, consider transparency. So consider how much data you can put out there and who you want to work with. So with that, I just wanted to thank you all and thank the organizers for letting me speak here about safety. And I just want to acknowledge the massive amount of people at New Harvest, at Vireo, my co-authors, and then so many participants, a lot of you who are here today who worked on the safety initiative. So thank you very much. Yeah. So yeah, you're right. I forgot to mention that we didn't sign any NDAs in order to do the safety initiative, and thanks to the trust that New Harvest has really developed in this community. I mean, that was a big, a huge contribution, bringing people together, understanding that they could trust New Harvest, and then also giving people lots of different venues to kind of talk about things. So the workshops, people were all on together. We were able to talk about things together. We gave people ability to give feedback anonymously in a lot of different ways. And then also the one-on-one talks where Isha spoke to the different companies. That was really a way that was an enormous amount of work on her part, and that was a really good way to move forward. We have done safety consortium before where we have signed NDAs, and so companies, we have experience with competitors working together in order to develop safety methods and be able to share some information with each other. But what we try to do is we often try to find representative products. So we use products that are representative of everyone's products, and then we do the testing on that, or we do blind testing. So that's some way to also, there's a couple different models that we could use for that kind of work.